Hello. Thanks for the kind introduction. Really happy to be here. Today I'm going to be talking about diet microbiome study design and using a few vignettes from some recent studies to look at the different ways in which diet does seem to interact with the microbiome and how that should inform the way we design studies. A couple of disclosures here. And all of the work that I'm going to be talking about today is all open source and peer reviewed. So I want to start with this concept. Diet is the main driver of human microbiome variation, and I like to pose this question to people and see what they think, because it's not actually entirely clear whether you should agree or disagree with this from kind of the sum of all of the microbiome literature. And if you look at various bits of evidence, you can see some strong evidence that yes, diet strongly dominates host genotype, but this is often in the absence of many other factors that are influencing the microbiome. When you look at human studies with dietary interventions, there have actually been a number of cases where even when people get very severe dietary interventions, they still retain their own personal microbiome kind of profile. They just move slightly within their personal microbiome cloud. So I want to walk through this case study that came out of my research lab recently, where we tried to answer this question. And then we'll look at how those findings inform how we might want to do study design. So this is work that was led by Abby Johnson, who's a postdoc in my academic lab at the University of Minnesota. And this was sponsored by General Mills. And Abby's got this great combination of skills, because she's a registered dietitian, she has a Ph.D. in nutrition, and she knows how to code and analyze microbiome data. So the first thing we decided to do is collect some big data. And that's what I'm showing you here. So this is a visualization of shotgun sequencing of daily microbiome samples from 34 people, sampled over the course of 17 days. Each of these boxes is one person, and within it you can see their microbiome varying over time. This is Abby's microbiome. She actually had the most stable microbiome. She was very proud of that as a registered dietitian. But on the other hand, you can see there's some others who are actually quite variable in their microbiome. And there are a number of other things you can see in the microbiome just by staring at this. For example, each person has their own microbiome fingerprint or profile. There are no two profiles that are the same. Some people are similar to each other. These three people on the right, for example, they're all Prevotella carriers. That's the red color. But nobody has a twin. So it seems that people are unique, which we know that from many other studies. And then, of course, because we're interested in diet, we also collected diet from the entire study, and that's what I'm showing on the bottom row here. And again, you can see the diet varying from day to day. And just by staring at this, you can already glean some interesting information. One of the things you can see is that diet is actually more variable from day to day than microbiome. And this is looking at diet at a very high level. These are like the sort of phylum level dietary features, whereas the microbiome, these are actually species level, so more specific characterizations of the microbiome, because we had that shotgun sequencing. And so you can see diet is more variable. And again, everyone sort of has their own ratio or mixture of types of foods. The other thing you might see is that there are these two cases in the middle here that are colored kind of pinkish purple that had something very interesting happen to them. And the way Abby put it to me was that it turned out they were computer scientists and had to drink only, or had to consume only Soylent for the entire period of the study. Apparently, this is something that's popular in tech circles, and I'm allowed to make that joke because I'm a computer scientist myself. So, so these two subjects had taken it upon themselves to switch their diet to only Soylent day in and day out for 17 days straight. It's like this meal replacement beverage. And what you can see is, very interestingly, even though it's only N of 2, they retained their individuality. If you look at their microbiomes above, they did not converge toward the same microbiome. So they stayed in their own microbiome cloud. And in fact, these two individuals were cohabitating during the study, and even still, they didn't converge. The other interesting thing is they really didn't stabilize. They might have been slightly less stable, or slightly more stable than others on average, but they retained quite a bit of variability from day to day that was a lot more than we would expect just from technical variability. So there's sort of an interesting embedded study here. Now, what we're going to try to do is answer this big question, how much of microbiome is driven by diet. And the way that we're going to approach this is actually to partition the microbiome variation into two components. There's the microbiome variation between people. So that can be thought of as the distance between the center of one person's microbiome cloud to the center of another person's microbiome cloud. Or the way we actually calculate this is it's just the average of all the pairwise distances from one person to another. And then we can do the same thing for diet, where we calculate the average distance between, say, my diet and your diet. And what we want to know is, are people who consume similar diets in the same space in the microbiome profile? And, if you have more similar diets, are you more similar in the microbiome? And that will let us look at this between person variation. Then, once we look at that, we're going to pull out the within person variation. So, as we started to do this, we realized we had no idea how to properly analyze the dietary data. We were looking at it, and it's incredibly variable, as I said. So here again is Abby's microbiome on the left, Abby's diet on the right. And you can see that even at this phylum level, it's far more variable than the microbiome. And so this was kind of funny to me because I spent the last decade complaining / bragging about how variable the microbiome is, and we have to deal with all this noise, and we need special methods. And now, diet is in fact far more challenging. So I found it quite fascinating. And actually, if you zoom in and visualize the specific foods in the diet, not just those food groups, it's even more variable. Now it's just like abstract art, and it's not clear at all how you would want to analyze this when a person is hardly eating the same foods from day to day. So we took a step back and said, let's just look at the diet data. And that's what I'm showing here. This is a plot that Abby made, a network diagram, where the white circles are the people, and then the little colored dots are the foods that they're eating that are colored by the food group. And the way this network works is it's trying to put the foods near people who are eating them and far away from people who aren't eating them. And then it also is trying to put people near people who eat similar foods. So you can see the two Soylent subjects are in the lower right, and they actually are connected to the center of the plot because one of them had a caffeine drink one time, and another one had I think a piece of pizza one time in the entire study. Otherwise, they would just be totally off on their own. So this let us really get a grasp of how sparse is this diet matrix, the food matrix. There were some foods, those are those foods in the center, that were eaten by a lot of people. And in this population, these were things like bread, for example. And then there were these other foods that were out on the fringe that were only being consumed by one or two people. And these were more rare foods that tended to show up in just one or two people. And what this showed us is that most of the foods, or many of the foods, were only being consumed by a small fraction of the subjects in the study. So the food intake was highly personalized. And what this means is that it's going to be challenging to analyze. If you picture this matrix of the foods or the people versus the foods they're eating, it's mostly full of zeros. You don't have the same foods showing up in different people. So how can you do any statistics with it? Well, this is where we start to think about methods for dimensionality reduction. And in fact, there's already this amazing way to reduce the dimensionality and project this sparse, very high dimensional space down onto a small number of features that are kind of like latent underlying factors that are in common across all people, and those are called nutrients. So we already have these for all of the foods in our food database. These are all the macro and micronutrients, protein, fat, different types of fat, and all of the vitamins and minerals. And this is actually how many of the diet microbiome studies have tried to compare dietary intake to microbiome variation, is using these, which makes sense because the foods are too sparse, as we just saw. So this is the first thing we tried. We said, okay, let's just take this nutrient table, and we want to take the average nutrient intake for each person over time. And then let's look at the average microbiome profile for each person over time. And now we can make two plots of the beta diversity, or the distances and similarity between different pairs of people. So we have on the left hand a plot of the principal coordinates analysis of microbiome, where each dot represents the average microbiome of a person. So people who have similar microbiomes on average are close to each other. These three in the bottom left-hand corner in red, those might be the three Prevotella carriers, for example. Then to the right of that, we have the average nutrient profile, principal coordinates analysis. And again, people whose dots are close together have more similar dietary or nutrient intake. And we're going to try to overlay these on top of each other. This is called Procrustes analysis. You might have seen this in some of the microbiome literature, and see how well they line up. And if we can get the red dots and the blue dots to line up, that means that people who have similar nutrient intake also have similar microbiomes. And when we tried this, in fact, it did not work, and we got kind of this hairball out of the analysis. And we tried this every which way. Let's take wet weight, let's take dry weight, let's cut out liquids entirely, different distance metrics and normalization techniques. We could not find really any way to get the average microbiome to correspond with the average nutrient profile. So we scratched our heads for a bit, and we started to realize, okay, this doesn't work, but maybe this makes sense because we're just looking at conventional nutrients. Like, the nutrients I showed you a couple of slides ago, those are what are shown on packaging, when you buy something at the grocery store. But those are the nutrients that we can metabolize directly for the most part. What it's not showing us is everything else that's in those foods that perhaps our microbes might care about enormously. And this is a really nice visualization put together by Robin Shields Cutler, who was also a postdoc in my lab at the time. That took this idea from there's a guy in Australia named James Kennedy who makes these posters where he shows the contents, the ingredients, so to speak, of whole foods. And this is one showing what's actually in a strawberry. And it's got all sorts of polysaccharides, different types of sugars. It's got coloring agents. It's got different fatty acids. It has preservatives, and various flavors. And these are completely ignored. All of these components of our food are completely ignored when we just look at these conventional nutrients. So you could think of these like the dark matter that's in our foods that the microbes care about. And this might explain why we have a hard time getting just this like very rough nutrient intake to correspond to the microbiome. Now, on the other hand, we have this problem because, as I showed earlier, the foods are way too sparse. We can't just use the foods directly. And fortunately, Abby had this insight that we could find some underlying structure that would help us share information between related foods. And what she noticed was that the food database that we were using was already organized hierarchically. So the foods had these food codes that went with them that showed basically, which foods were in the same general type of group. And she used this to derive a kind of pseudo phylogeny, or a tree that organized all of the foods. And it's not perfect. Certain things might be complicated if there's like, say, a dish that had some grains and some meat in it. But at least it gives us the ability to pull some information out of these foods that would otherwise be treated as like completely separate features. So with this tree, we can now do things like compare my dietary intake to your dietary intake, not based on whether we ate the exact same foods that day, but on whether we ate foods that were near each other in the tree. So if you and I both ate like a mixture of beans and vegetables, that would be a sign. Hold on, I've got a alarm going off here. That would be a sign that that we were more similar to each other than to someone who had eaten like a mixture of grains and meats or eggs. So now we turn back to this problem of comparing microbiome between people to diet between people. But here we're taking the food tree unifrac distances between people's average food intake profiles. So unifrac is this very powerful beta diversity metric that's been very effective for comparing microbiome samples. Now we're able to apply it to diet. And when we take these two clouds and line them up, they actually lined up significantly. And this approach was the only way in which we were able to get a correspondence between a person's average microbiome and a person's average dietary intake. So, to me, this was a little bit of evidence that that the food tree is sort of working, and that it might be helping to capture a little bit of the information about this dark matter in the foods. That ideally we could just have a full profile of all of the chemical constituents of every single food, but we're a ways off from that for now. So this is a way of kind of getting around that, while we only have these very rough conventional nutrients for each food. And from this analysis, we can see that the foods explain about 20% of the variation between people in their microbiome. So is diet the main driver of microbiome? Well, maybe, so far we've only been able to explain about 20% of microbiome variation from diet. But we've left out all of the variation within a person's cloud. And that's what we're going to look at next. So now the question is, let's take one person, look at how they're bouncing around in their own microbiome space. They might become different from themselves for a few days, and then they might move back to where they were, for example. And let's compare that to what they've been eating recently and find out whether your recent dietary intake helps explain today's microbiome, your microbiome today. And for this analysis, we'll do the exact same thing. So we're going to take the principal coordinates analysis of one subject's microbiome. So this is all 17 days from subject 36, for example. Then we'll take the same thing for their dietary intake using unifrac distances on the food tree data and try to line them up and find out if they correspond. And then when we get a result, we get a p-value. And because it's a time series, we thought it would be kind of interesting to visualize it as I'm showing on the bottom of this slide. So basically, the first axis of this Procrustes analysis plotted against time. So when we plot this, if there were no correspondence between your recent dietary intake and your microbiome today, then these points would be flip-flopping up and down, from the top to the bottom of the graph. The blue and purple, which represent microbiome and diet, would not be moving from one end of the PC1 to the other together. And so for this subject 36, we actually see a strong correspondence, and there's the p-value. And now I'm going to show you the other 28 subjects that were significant. So these are like the top 24 subjects, and you can see like there's a strong correspondence across many of them. So and this allowed us to ask the question now: how much of the variation in microbiome within a person over time was explained by the variation in diet within a person over time? And the diet explained about 24 or 25%. And I say diet explains greater than or equal to 24.4% because I know that we could explain more if we had more accurate ways of collecting dietary intake. So that's kind of like a floor for the value to me. So I just think of this as 25. And so now we know that diet's explaining 20% between people and 24 and about 25% within people. And there's a very interesting finding that came out of this analysis that I'm going to share with you now that I think has some important implications for how we might want to collect these types of data in the future. What I'm showing here is on the y-axis, the vertical axis, the significance of one person's or each person's diet microbiome association over time. And it's the negative log 10 of the p-value, so higher is more significant. And then on the x-axis is how many days of dietary history would be used, because we could just take yesterday's diet and say, that's what we're going to try to correspond with microbiome. Or we could take the last 2 days, or we could take the last 3. And what we found was that on the left hand, where we only used 1 day of dietary history, we had relatively few subjects with a strong correspondence. And another way to say that is diet did not really explain their within-person microbiome variation. It wasn't until we took 2 days so this is just taking the average of your last 2 days of dietary intake and tried to correspond that with today's microbiome that we started to get the majority of people having a significant correspondence. And then, we also tried 3 days, 4 days, 5 days. it didn't really change much. Then one of the reviewers on this paper suggested that we try a model with a decaying influence. So let's make yesterday worth the most weight, and then the day before is a little less, and the day before is a little less. And so we just used like, 1/2, 1/4, 1/8, and so on. And when we tried this decaying model, that's when we actually got the most significant associations, where we have all but 4 of the subjects having a significant correspondence between their daily diet and their daily microbiome. And there are probably some questions that are, you have popping up, like, is this just because of transit time? Maybe you should take just 2 days ago because food takes 2 days to get through the gut. Well, we tried that, and we didn't get any stronger signal just looking at only 2 days ago or only 3 days ago. I'm sure there are transit time effects. Some people, have a rapid transit time. Some people have a slow transit time. We don't really have that information here. So, it might be that we can actually do a better job of explaining if we were to incorporate that transit time and have kind of a different offset of history for each person. But then again, the weighted average probably wouldn't be expected to be more effective if that were the only thing driving this history. So I think, it's a combination of people have different transit times, and also, it takes time for the effects of what you eat today to get processed by or kind of reverberate through the microbiome and have kind of like secondary and tertiary effects that take some time. And so, this was very interesting that it really mattered having that dietary history. And this is something else that might be getting in the way of some microbiome diet studies. If we only consider yesterday's diet, if we only ask people, what did you eat in the last 24 hours? That may not actually tell us much about, how diet relates to the microbiome. We need more dietary history. The other interesting thing that we were able to do is, because we had longitudinal data, we were able to ask whether a particular food was associated with a particular bacterium in each person. So we can drill all the way down and find out if there are personalized diet or food microbe interactions in each person that may or may not be the same, for different people. And we actually found many of these, dozens of these associations. And there were some that were more generalized. So on the left here, we have people who ate foods in this category, cakes, cookies, bars, recently tended to have more of this bug, Roseburia inulinivorans. And this kind of makes sense. Look at the name of that bug, inulinivorans. So this means it eats inulin. Inulin is like a very common fiber additive that's used as a bulking agent and additive in a lot of like bars and things like that. So, maybe there's some plausibility here. On the other hand, there were a number, actually the majority of the associations we found were not personalized. And these are just a few examples. The more dark green vegetables the subject 18 ate, the less Bacteroides species 116 they had. But, a couple of other subjects, on days where they had recently eaten a lot of dark green vegetables, they had more of that bug. Similarly, recently consuming a lot of pork made this Bacteroides oleciplenus go down in some and go up in others. So this is very interesting, and I think I'd love to see the confirmation of this in a larger study, but it's in line with a lot of the recent findings around, personalization of how diet impacts glycemic response through the microbiome, or how, probiotics might engraft in different people. And I think it really adds a new dimension to how we think about diet, microbiome, analysis, diet microbiome interactions, because there are different reasons why this might be happening. One of the possibilities is that it's just strain level differences. So, like, okay, my Bacteroides species 116 is actually a slightly different strain than your Bacteroides species 116, and they have different metabolic capabilities. That's why they respond differently to the same food. I marked that as easy because we can do really deep shotgun sequencing and assemble the genomes and find out, what the strains are. We could also culture the bugs from different people's guts and compare them. Another possibility is that it's food level differences. So my spinach is not the same as your spinach because it's a different strain of spinach, or they were, mine was steamed and yours was baked or something like that. And, or maybe, one of them had different types of soil microbes on it. So I call this medium because it is in theory tractable. We can control diet. We could have complete control over diet if we want to. It's just burdensome. So this is something we could study and measure. And then the last possibility for these personalized diet microbiome interactions is ecosystem differences. And I call this hard because this is where, even if you and I have the same strain of Bacteroides 116 and we're eating the exact same spinach prepared in the exact same way, the rest of our microbes might be different. And that strain that we each have of Bacteroides has to interact with the rest of the microbiome. So, it might be that the Bacteroides is actually responding to how other microbes are degrading or consuming the spinach, and in a competitive way. And I called this hard because it's so complex. Everyone has a different ecosystem, and we don't still really understand the rules that govern these ecosystems. I mean, at the very, very basic level, it's all physics, which means at the very, very basic level, it's all math. But we're a far cry from knowing all of those equations. What are all of the substrates? What are all of the enzymes? And which ones are in which bugs and which ones are in which foods and so on. And I think in reality, it's probably a combination of these three. So we have quite a challenging problem if we want to really solve this in kind of a reductionist or like bottom up sort of way. But the good news is we can do longitudinal studies in people and find relationships like this. The other interesting thing here is, if this is true, that food microbes interactions might be opposite in different people, then this would explain why we've had trouble finding strong relationships between foods and microbes in large cross-sectional studies. Because, if pork and Bacteroides oleciplenus are going up in one person and down in another, it doesn't matter if you have a study with 10,000 people in it. You still won't uncover any association between these because it's different in different people. They're canceling each other out. So this shows, I think, that you really do need to do temporal studies, longitudinal studies, and you need to be expecting some kind of personalized component that might actually be getting in the way of our diet microbiome understanding. The other interesting thing that we can do with this longitudinal data is, there are a lot of interesting things, but we can measure the stability of each person. And the way we characterized this in this study was basically like your average change from day to day, or what's your average distance to yourself in the microbiome space. So that's your microbiome stability, or it's actually the inverse of your distance. And we can find out if that's associated with anything in the diet. And one of the things we uncovered was that there was a very strong association between a person's microbiome stability and the average diversity of the foods they ate recently. So this means basically people who tended to choose a wide variety of foods on the food tree tended to have more stable microbiomes. And this was a very strong correlation of 0.52. For like an unstructured study like this with self-reported diet with no control of the dietary intake, I was quite surprised by this. So I think it might be a real association, and there could be some ecological explanations for it. And this is a new finding that came out of the study that we would not have been able to observe without that pretty dense longitudinal data. And we do know that stability is associated with various diseases that are related to the microbiome, or rather, lack of stability is associated with various diseases. Here's another interesting finding, and this is not something that was in the publication, but I think it's something that's again quite powerful and tells us a lot about what we should do in the future. So what I'm showing here is the diet microbiome correlation, or the percent of microbiome variation explained by diet on the y-axis, and then how many consecutive days of microbiome did I sample. And this is the correlation of diet microbiome between people. So this is where I'm taking your average dietary intake and correlating it with your average microbiome. If I take, so I have all 17 days for each person in this study, but if I subsample and pretend I only had 1 day, I can find out how much power I would have, or how much of that association I would be able to detect. So that's this first point. And what it shows is if I only had 1 day of one microbiome sample, even if I had this very robust measure of diet, because actually I'm using all 17 days for the diet, I can still only detect a fraction, maybe 75% of the full signal. And there's a bit of noise there if I try this repeatedly. As soon as I go up to 2 samples, so this means 2 consecutive microbiome samples, say Monday and Tuesday, and take their average, all of a sudden I get this big boost in the power to detect this signal. I can now detect 18.5% or 0.185 correlation. And then I get another boost once I get up to 5 and maybe a little boost as I get up to 7. And so this is something that actually was shown recently in another study, that if you collect multiple days of microbiome and average them, you increase your power quite a bit. And this maybe is kind of obvious because you have, if you're doing a cross-sectional study, so this is not necessarily even a longitudinal study, a cross-sectional study, and you want to find out a cross-sectional study, and you have several microbiome samples and average them per person, you're averaging out the kind of daily variation or temporal fluctuations in each person. And that's what's, you're getting rid of noise, so that's what's increasing your power. So I think what this means is that, like, coming out of this study and looking at some other data, when I design studies now, I always try to collect several consecutive days of microbiome, even if I'm not planning to do a longitudinal analysis, just because I want to average out all of that, day-to-day noise in each person. So that's something else interesting that came out of this study. This is a kind of summary that came out recently that Avi led. There was a collaboration with Angela Zivkovic that showed, basically tried to walk through some of the things I just showed you, from that previous study, and also made clear what we think are like the best practices moving forward. So I encourage you to check that out if you want to see all the gory details. But I'll give you a little summary of that now. This is an example of dietary intervention design where we're trying to test, dietary intervention and assess what the microbiome is before the intervention and after the intervention. And what we find is that we actually want to get several microbiome samples before we start the intervention, just to take their average and get a good understanding of the baseline microbiome. And then again, at the end of the intervention, we want to collect several consecutive microbiome samples and take their average. So we're only using them as kind of a single time point, so to speak, but having multiple of them will get rid of all that day-to-day variation in each person. And you might think, I've heard people say like, well, it's a lot to ask people to collect multiple microbiome samples, but it's actually a lot less burden, overall, to ask the existing subjects to collect a few extra samples than to go recruit a whole additional subject, which is the other way to increase your power. So I think this is a much more efficient way to increase power. And I've found in a couple of studies, Abby and I have seen that people they don't seem to mind collecting consecutive samples. In fact, like once they get in the routine of it, it's almost easier to just have them collect consecutively through the whole study. And, rather than having them start and then stop and then start and then stop, where the compliance might be a little bit lower. They just sort of get in the habit of it. And then you can always freeze the samples and, worry about which ones to analyze later. And then, so that's in this diagram, that's what these little M's are. Those are the microbiome samples before treatment and after treatment. But then you'll notice the D, those are the diet samples. So diet samples, we start at least a few days before collecting the microbiome, and this is getting 24-hour recalls of diet, so that we can incorporate that dietary history before the microbiome samples. And without that, again, as we saw earlier, there's so much noise from day to day in diet that we're losing a lot of the signal. We really have to have a few days of diet. Or, you can do a food frequency questionnaire. Those have their own issues because they don't tend to cover all foods. But, if you can't get multiple days of diet, that's another good way to handle this. I won't go through all of these factors, but in this paper, we walk through various potential confounders or factors that would be influencing the microbiome that you will likely want to control for in your study. And I just made this little point here on the top right. When possible, I like to control for these through the criteria and the sampling frame of the study. So if you can just exclude people who have certain confounding exposures, that will be a much more powerful study than if you include people with those confounders and then try to kind of regress them out after the fact using statistics. Even though I'm a data scientist, I'd rather solve things upstream of the statistics. That's a more powerful way. And so, that's just a little bit of a list. You can look at it in the paper if you want to dig in more. And then, we also have this table of recommendations that are divided up into what we think is like feasible to do right away in pretty much any study, if you can get like sufficient budget for it. Then we have what's ideal to do now. So it's like technically possible now, but might require a little bit more like resources and dedication. And then future needs, this is where we really need to do future work to improve the methods that are available to us. So in this first category, which is just the study design, how you design the sampling, you really want to increase your sample size for cross-sectional studies. You can do that now, and you really should be collecting multiple fecal samples per study time point. Then, what's ideal is actually to do dense longitudinal sampling during and after intervention, so that you can do a longitudinal analysis within each person, and maybe collecting even more fecal samples, like up to 7 we saw, and another study found that recently too. Up to 7 samples consecutively can really still improve your signal. So those are some thoughts on what you can do now. For microbiome design and data collection design. For dietary assessment, food frequency questionnaires are okay. They don't hurt, but they don't always represent everything people are eating. We found that we really increased the power getting multiple 24-hour dietary recalls. And we need these for 2 reasons. One, because the dietary history, the last few days mattered with the microbiome, but it also gives you a better sense of a person's habitual diet or average intake. As you start to get more and more 24-hour recalls, you start to get a better focus on the centroid of their diet cloud. And this is a little bit burdensome. I have done these 24-hour recalls, and they take 30 minutes a day, which is a pain. So we do need better tools here. But if you don't get a few days of history, you're going to have a harder time finding associations between diet and microbiome. So it's sort of a necessary evil. Ideal now, is to have participants report their food intake with multiple 3-day records paired with each microbiome sample. So basically, you always have at least 3 days of history, dietary history, for every microbiome sample, and that would allow you to best remove confounding effects of diet, or look for dietary interactions. And you also want patients to really be trained on how to collect their diet, and preferably have a dietary professional do this, have someone following up with them during the study, making sure they're understanding it, checking to make sure that they're not claiming to eat 400 calories a day or 4,000 calories a day, and making sure the data are high quality. And then if you're going to do dietary interventions, we had a few recommendations. I think the most salient here is something that you could do now is to think about, if you're going to do a dietary intervention, I think some people have a tendency to want to standardize the diet before they do their intervention. So let's give each person the same diet, and then we'll change something about their diet. And the problem with that, you might be able to think of the problem with that for yourself. The problem with that potentially is that the microbes in each person are not used to that standard diet. They're used to that person's habitual diet. So the very fact that you gave them a standardized diet is causing them to, that in itself is an intervention. It's causing their microbes to have to respond. So we think it's going to be more useful to instead stabilize their diet, find out what they've been eating habitually, and ask them to retain that diet and not change it over time. And then we have some recommendations for dietary analysis that you could dig into too. But really, it comes down to some of the things I showed earlier. Just looking at these conventional nutrients is probably not going to cut it for microbiome. So we should be looking at specifically foods and food groups, and maybe dietary patterns or healthy eating indices. And if possible, having longitudinal analysis is good, and doing something like the food tree, where even if people are eating slightly different foods, you can somehow use that information to get more signal out of the data. So I'll let you dig into this paper, if you want to read more about more of the details on the recommendations and what some of the future needs are. And with that, I'd like to conclude and thank you all for your attention. Thanks for the opportunity to present. And thanks, of course, to people in my lab, past and present, who've helped out with a lot of these findings and studies, and to some of the funding agencies who funded this work, as well as my other collaborators. And thank you so much. I'd be happy to take any questions.
Excellent. Thank you for your presentation, Dan. As you just said, it's now time for the Q&A. To ask Dan a question, just type it in where it says ask a question and then press submit. So Dan, our first question, this one asks, how should I control for a habitual diet in a diet intervention study?
Ideal would be if you can very accurately find out what people are actually eating day to day for several days, preferably a week or so, because that's giving you a real sense of what their habitual intake is, rather than just asking them to recall in a food frequency questionnaire what they think they eat habitually, because they're not necessarily exactly the same. And so that's what I would do if I could design the study any way I wanted.
Excellent, thanks, Dan. Our next question asks, can a baseline be achieved for an individual who's fasting? Is that something you can help answer?
I think that would be a very interesting study. How long do you need to fast before your microbiome stabilizes? I mean, the problem with fasting is that it's sort of like a continuously increasing dosage as an intervention. Like, the more days you fast, the more severe or strong, the so to speak, dose of the intervention is. So I'm not sure if you would, I don't know if the microbiome would ever really stabilize as you fasted for more days, but I think you would see a stabilization if you had intermittent fasting, for example. So, if someone's fasting Mondays and Wednesdays, and they do that for a month or two, at a certain point, you're going to start to see the microbiome stabilize. I don't really know how long that would take. That would be a very interesting question to answer. But yes, I mean, I think it is possible. You just need to do a longitudinal study with enough people to really get a sense of when does it stabilize? How long does it take?
Excellent. Thanks, Dan. Our next question asks, why use Aitchison's disease for microbiome? I think that's principal coordinate
Aitchison's
analysis.
Right. Yep. Yeah. Aitchison's distance for the principal coordinates analysis. Yeah. So Aitchison's distance is a distance metric for log transformed or centered log ratio transformed data, where it's a transform to the data that gets rid of a lot of the artifacts that come from compositionality, which means the data are percentages. You can say your percentage of a certain bug went up, but you don't know what the absolute magnitude of all the bugs was. You don't know the load or the biomass of the bugs in each person. That might be changing too. So Aitchison's distance basically means, you do a transform of the data. We did this centered log ratio transform, and then you calculate Euclidean distances on that, which we found is a very effective way to compare microbiomes. We used that instead of Unifrac on the microbiome, for example, because these were shotgun data, and we didn't really have a good tree at the time relating the species and the features to one another. So we had to use something that was not informed by phylogeny. We could have used Bray-Curtis, that worked fairly well too. We could have used other distance metrics, but this one, Aitchison's, we found is quite powerful.
Great, thanks, Dan. Our next question asks, What if this study was done with people that were on the same diet? So, for example, prison cellmates, for instance.
Yeah, same diet is very interesting because it would really, especially if they're on same diet for a long time, because it would really help you see like what's left over in between person variation. I think you'd still be able to see if there were certain personalized effects within each person. That would be very interesting. Two people eating exactly the same thing over a long period of time, are similar microbes changing in similar ways or different ways? But I think you'd really have to think about the rest of the exposures. So if you have, for example, prison cellmates or inmates, they wouldn't generally be having, I don't think, the same types of variation in external exposures that we would expect in the general population. Moving about, going to different sites, visiting different geographical or different buildings, and being around different groups of people frequently. So you'd learn something, but you'd have to be careful about generalizing it to the general population because of all of the other exposures also influencing the microbiome.
Excellent. Thanks, Dan. Our next question asks, have you looked at the KEGG data to see if the functional differences and responses may be more conserved than the abundance differences?
That's an excellent question, and I think an excellent idea. And it is something that we looked at in those data, and we didn't really find strong conservation there either. Which I think really, it's obviously, as I said, underlying everything. At a certain point, we should be able to get down to what are the enzymes present in these bugs and how are they interacting with substrates in the foods. So I think it's probably, just that there are, issues with like knowing exactly the food preparation, and the collection of the food data, as well as not knowing all of the genes. I mean, still many of the genes that are in many of these bugs that are going up and down are just completely uncharacterized. So I think, there could be some associations if we zoomed in and just looked at like short chain fatty acid production, maybe we'd see something, but when we just took all the enzymes and compared them with all the foods, all the known enzymes, I should say, we didn't find anything. A few signals, but nothing really compelling.
Great, thanks, Dan. Our next question asks, have you considered doing a stool test to find out about possible pathogens present in the gut that could explain the variations?
I mean, we did test for whether specific bugs were associated with specific foods, and there weren't pathogens, obvious pathogens coming out in that analysis. We also tested for whether there were specific bugs associated with stability. And there, in fact, we did find a number of bugs that were strongly associated with an individual's microbiome stability. But, again, no really strong, obvious pathogens jumping out there.
Great. Thanks, Dan. Our next question is in relation to the repeated sampling affecting power slide. Does it matter if the sampling is in consecutive days versus once a week during several weeks?
Ooh, good question. So if you're trying to assess baseline microbiome and you collect it over the course of several weeks, that's probably fine because presumably there's no like change in behavior or intervention taking place. If you go back too far in time, you're going to have some drift of the microbiome, which we know happens. So Now, I wouldn't stretch it out too far, but probably, weekly for a few weeks is okay. If you're looking at post-intervention, then it's not really a viable option because you're trying to assess the state of the microbiome right at the end of the intervention. And if you draw it out over the course of a few weeks, the effect of the intervention might be wearing off. So I think it depends which type of study time point you're talking about.
Thanks, Dan. And our next question asks, patients with inflammatory bowel disease, do you think the microbiome could be used to predict the responses to treatment? And I suppose that it's similar to other things. Can the microbiome just be used as a biomarker in general?
Absolutely. I believe that this is the case. We have mounting evidence in a number of different areas that microbiome can predict response, and that could be in several different ways. So we know that the microbiome is involved in metabolizing a wide range of drugs or pharmaceuticals that are used to treat various diseases associated with the microbiome. And this could be making the drug more active. It could be making the drug less active or efficacious. It could be making the drug more toxic and causing you to have to reduce the dose. So that's one possibility where I think it could predict the response. There's also been a strong signal in immuno-oncology that using immunotherapy for certain types of cancer seems to be more effective in people with a diverse microbiome and maybe a certain type of a microbiome, which might have more to do with how the microbiome is interacting with or activating the immune system, or maybe it's just informing you about the kind of status of the immune system. So I think those are at least two ways in which microbiome seems likely to be able to predict response to treatment in a few different diseases.
Excellent. Thanks, Dan. Well, I'm afraid that is all we have time for today. If you did ask a question that we didn't get time to ask, Dan will try his best to get back to you offline after the webinar. I would now like to thank Dr. Dan Knights for his presentation and for answering your questions. And of course, you, the audience, for taking the time to be with us today. Thanks for watching, and I hope you'll join us again soon.